All right, I hope that you guys enjoyed that nice little break. The chocolate things are pretty darn good. So, um, so next up, we're going to have Luis Alvarez come up. He's the founder of Theradaptive. Luis is a graduate of the U.S. Military Academy at West Point and served 20 years on active duty with the U.S. Army. While in active duty, he co-founded and led the DoD Regenerative Medicine Program Office, which is very impressive. As he came to MIT as a Hertz Foundation Fellow in 1998 and first completed a master's in chemical engineering with Charlie Cooney. He then went on to do his doctoral research with Linda Griffith and Richard Lee in biological engineering, studying cell behavior with engineered HER receptor ligands for regenerative medicine. Luis is the founder of Theradaptive, which is developing a therapeutic delivery platform for multiple orthopedic clinical indications, including spinal fusion, trauma repair, dental repair, and complex cartilage repair. Please welcome me in joining Luis.
Oh, and to flip back is red. Got it. Okay, thank you. I like to test the equipment before I use it. It's a lesson from the military. Anyway. A pleasure to be here again. I want to thank the organizers for really putting together what is a great tribute to Danny's life and work, and it's an honor to be invited, so thank you again for the opportunity to share what we're doing. In preparing for this, I dug through some old pictures that I had and came across this picture here. This is from 1998, and picture there, myself and Bernard Schilling, who was a postdoc at the time. And there we're shown, quite happy with ourselves. We had just resurrected B. Braun fermenter and brought it back to life, and I started doing work in microbial desulfurization, which Christelle had talked about. So we were very pleased with ourselves. We had also taken the time to read Charlie's thesis, his master's thesis, and knew that we were going to have to remove heat from this fermenter as it grows its culture. So we connected the fermenter to the house line and let it rip. And we went off to, the thirsty ear or the muddy to have a beer. And the next day when we came back, the lab was completely flooded. So although we had remembered our chemical engineering principles, we forgot our plumbing. So I had to go and offer my mea culpa to Danny, and his expression did not deviate from this here. So he handled it very well and taught me how to handle kind of disaster. So Charlie, I don't know if you knew who was responsible for it, but it was all Bernard's fault. So as was mentioned here, I served in the military, and between my time doing a master's and then coming back to do a PhD, the Army required some additional service. And I was deployed to Iraq, and while I was there, I started hearing reports of what's called delayed amputation, essentially where modern military medicine had progressed to such an extent that you could salvage a limb that had been injured due to an explosive event, for example, or trauma, but that weeks or months later, that person would have to have an amputation. And that to me seemed barbaric. Why is this happening? And in digging further with some of my friends at Walter Reed, learned that really this was due to the fact that really we didn't have the ability to guide the regeneration of certain tissues that would preserve the viability of that limb long term. And that includes tissues like bone, of course vasculature and others. So this provided the motivation, and when I came here back to MIT, there was a new department called biological engineering. Of course Doug was kind enough to let me in, and Linda was kind enough to host me in her lab and serve as my advisor. So the technology I'll talk about today was really developed in Linda's lab. So as a company, we're focused on the area of precision tissue regeneration through the delivery of therapeutics to very specific regions within the body. And as a business model, what we've done is taken particular therapeutics that maybe have a specific clinical indication, show compelling data, and then form partnerships to expand the platform. So we get emails like this all the time from people. Although the original motivation for our work is in combat trauma, there are actually many, many times more people that have delayed or non-union injuries, and these invariably lead to one of two choices. Either you are going to remain bound to a wheelchair or not able to have mobility, or you choose amputation. and do your best with the prosthetic. So it's a choice that no one should really have to be faced with, and the work that we're doing, we hope will eliminate that dreadful decision. So, if you look at the history of how therapeutic interventions usually work, either you have devices, which are hardware, and these devices normally do not have any biological function other than the mechanical support they provide. And then you have biologics, which have potent biological activity, but which are formulated for systemic delivery. So it's very hard to control in a targeted way the delivery of that biologic function. So we operate at the interface where we combine biologics with the precision placement of a hardware implant. So our platform is based on the idea that you can modify recombinant proteins in such a way that they gain an important function, and that is to adhere very tightly and very specifically to certain classes of materials that are present in implants. The way we do this is we have a combined approach that uses both what you might call traditional, high throughput screening methods for protein libraries, such as phage display methods, and then a computational approach to take those enriched, you might call them evolved or identified sequences, and then further refine them to make them suitable for use as part of a fusion with a therapeutic. And so what you end up with is a variant that retains its original biological function, but now has gained the ability to bind very specifically and with very high affinity to certain classes of materials. What's interesting about this is that it's portable to basically any recombinant protein you wish to make. We've made over a dozen of them using these types of modifications. And you can restart the IP clock on that molecule because it's now its new composition of matter. So, a lot of the therapeutics that are out there that are biosimilars really cannot rely on enhancements to the composition of matter. So what is the basis of this approach? One very important thing to remember is that any material, well, I should say many materials that are used in implants actually exhibit what we call periodicity of material properties because they have a crystal structure or a mineral structure. And so we take advantage of the distribution of these pockets of surface character to allow us to grab onto something with a peptide side chain. And so when you're screening against materials, you're really exploiting the specific interaction between an amino acid side chain and these pockets of character. And it can be charge, or it can be the solvation of water, which provides hydrogen bonding opportunities, or hydrophobic patches. So you have a lot of real estate happening, a lot of interesting topology happening on the surface of these materials. So one approach that we take is we say, look, you can take traditional phage display approaches, for example, and you get sequence information for candidates that may be useful in this regard. But usually those sequences are not optimized for other things that you will need. For example, when you go to scale up a therapeutic, you need to be able to express it at high levels. It needs to refold. It cannot interfere with the biological function of the fusion protein and so on. So we've developed a type of computational platform that complements or extends the utility of a phage display approach. So then you can begin to formulate your approach to making recombinant proteins that have these tags that confer very specific and very tight binding to materials without interfering with other functions, and that also help to optimize the expressibility so that you can achieve true scale-up. So how does this work practically? Here you have an example of an implant. This is characteristic generically of an orthopedic implant, which can be used either for trauma repair or for dental repair, or even for more importantly, for spinal fusion. Those implants are inactive, biologically speaking. They just provide a substrate for cells to lay down matrix. With the technology we have developed, you can take a type of implant like this and in a one-step dipping process, completely quantitatively deplete the protein from solution and have it become deposited on the surface to create a bioactive surface. So, in contrast to encapsulation or time-released approaches, which do not make the molecule available, these are presented on the surface, and so any cell migrating over that surface is immediately activated by the ligand that's been bound. So this provides an interesting and very scalable manufacturing platform to make bioactive implants, which normally would take very complex chemistries or post-processing. So as a platform, this now is interesting because you can say, well, what clinical indication do I really want to go after, and what implants or devices have traditionally been used to address some of those conditions. None of those, if you look across the market, none of those really provide biological function. Now you can ask the question, what biologic would I want to load onto those implants? In a sense, I like to use the analogy that now you can load biological software onto medical hardware. So now you can convert these implants into a next-gen type of device that actually has a biologic function that you would desire. As a company, we have focused on 3D printed and trauma, or what we call trauma repair implants, and then the larger segment, which is spinal fusion repair. So of course, you have a platform, and as a small company, the first question that you're faced with is, what do we do first? So we really studied the market quite a bit to see what opportunities there might be to disrupt what is going on now and to fill a gap that is quite needed. And after doing that, we arrived at a molecule called BMP2, or bone morphogenetic protein 2, which is a very potent inducer of bone formation. You can essentially place this molecule under a muscle flap or in a defect, and you will induce bone wherever that molecule goes. Unfortunately, it's very hard to control where you deliver that molecule, and essentially you end up with off-target effects, which can be quite devastating if used in the spine. So we applied the platform to modify BMP2 and come up with a proprietary variant that is has a very high affinity for calcium phosphate mineral content. And we call this new molecule AMP2. So AMP2 retains all of the original bioactivity of BMP2, but now has the ability to bind with extremely high affinity and specificity to materials that contain calcium phosphate, such as hydroxyapatite and beta-tricalcium phosphate. And what we have found is that, well, of course, 80 to 90% of the implant market in orthopedics and spine repair actually contains implants that have tricalcium phosphate or hydroxyapatite. And so we've been able to show that you can actually load a potent bone-inducing biologic on every device that we've tested so far. We've tested about 100 or so. So that has created quite a bit of interest in the market. And we've been able to establish partnerships with two companies who are interested in converting their existing implants into biologically active ones. But of course, the number of different devices here, there's quite a bit of diversity. So you can do anything from implant strips, ceramic-coated metal, or electrospun materials. So the number of applications really becomes quite large. So I want to show you some results from this type of approach. And I'm glad we're doing this after lunch because it's a little bit gory. But essentially, we wanted to test the hypothesis, does ultra-precise presentation and localization of an implant with AMP2 lead to better outcomes in the context of bone regeneration? So in this model, you produce a very large segmental defect in the hind limb of a goat. And this defect is about 5 centimeters. So on its own, this would never heal. And you would end up with what is called a non-union. And this invariably leads to amputation. But to make the model even more challenging, you place a spacer and delay the healing by about a month. And then you place your test article. So in our case, we partnered with a Japanese company that makes an electrospun composite ceramic material, kind of like a wool. And we're able to load AMP2 onto it very effectively. And you can see what it looks like when it's placed in the defect. So for surgeons, it's very easy to use. The other aspect here is that the end user, who's going to be the surgeon, needs to be able to manipulate it to fit the wound geometry. So what do we see? At the beginning, when you first create the defect and then remove the spacer, you see no regrowth of bone. And in the control condition in which you have just the naked wool implant, you also see no bone at the end of the test period. At the end of 12 weeks, however, in the AMP2 case, we see complete regeneration of that defect to such an extent that you really don't know where the defect was to start with. When the surgeon first saw this, he had to look at his tech and said, where did we actually cut this thing? So he was very impressed with the final outcome of this. And not just once, but several times. So in every case where we added AMP2, we're able to demonstrate a very controlled, anatomically correct regeneration of the tissue, which is the holy grail in tissue engineering. So we're very pleased by this result and have also conducted a number of other studies in spinal fusion that have shown the same result. Very precise, controlled bone repair without any off-target effects. So building on that, we started expanding our work a bit into the creation of anatomically shaped implants that can be used for personalized repair procedures. And this becomes more important in cranio-maxillofacial repair, where you have anatomically complex shapes, and you may not want to rely on kind of a more of amorphous type of implant. So we developed a material category here that we call flexible ceramics. It's actually 80% ceramic, but behaves like a composite. And we've been able to partner now with 3D Systems, which is the world's largest 3D printing company, to augment their existing workflow. They print custom surgical guides and metal implants for orthognathic surgery. So this is correction of the jaw and craniomaxillofacial bones in cases where you have some either disfigurement or a congenital defect. But what they were missing is what to do when you are left with the gaps that are caused by this procedure. There's no way to regenerate that bone easily. And in many cases, you do not get healing, which leads to disability and long-term morbidity. So we've partnered with them to develop now what are customized, or really the world's first personalized orthopedic regenerative implants. And shown here in this concept, you can backfill any gap that you might create, and you would get very precise regeneration. I'd like to pay tribute here actually to Charlie Cooney, who serves on our Science Physician Advisory Board and has actually kind of helped us quite a bit on the protein therapeutic aspects and the scale up. So his impact is being felt even to this day, even though this was many years ago that I flooded your lab. Apologies for that. And also Linda Griffith, who is our academic co-founder. This work came out of her lab, and actually she created the environment that allowed us to explore this. This was initially not the original subject of the thesis. It was a kind of a side project. But she was kind enough to say, go ahead and give it a try and see where we can take this. So she created an environment that really allowed this type of idea to flourish. So I want to thank her for doing that, and hopefully she won't hit me with a hard question. And again, I want to thank the organizers. This is really a great way to pay tribute to Danny's work, and happy to take any questions.
Thank you. That was beautiful, exciting work. Opening the floor for questions. Oh, we have a question here in the back.
Hi, Louis. Thank you very much. I'm curious what the inside of the bone looks like after it's been healed or repaired in that manner.
That's a great question. Actually, that's one of the most important steps is to cut and perform histology on that. And actually, the bone remodels in a very physiological way, in that it's driven by the loading that bone experiences. So in the extremities, you'll get cortical bone forming the outer shell. The cortical is a much more dense form of bone. And on the inside where the marrow space is, you get trabecular bone. So you get more of the vascular ingrowth that's possible through the marrow. So a lot of that is driven actually by the physiological and mechanical loading of the limb. In the craniomaxillofacial space, where you don't really have as much of the loading signal, you end up with a trabecular bone that looks similar to the mandible next to it. But that's a great question.
All right.
Very simple question. Do you use the exact same technology to feed the protein regardless of what the surface is?
For a given class of material, we've been able to find that, yes, you can use the same fusion partner. So for all classes of calcium phosphate mineral, alpha and beta phases, hydroxyapatite, and even fluoroapatite in the teeth, you're able to get a very good binding. But as soon as you change to a new class of material, you have to reapply the screening process. Yeah.
Wonderful. Thank you so much.
Thank you.
Please. All right, very exciting. So our next speaker is Tasos Yanakakos, who's will be with us virtually. Tasos was an MIT undergraduate where he graduated in 1994 with a dual degree in chemical engineering and economics. He then went on to Northwestern, where he received his master's in biotechnology before coming back to Cambridge to do his MBA at Harvard. Tasos has been the founder of 3 companies and is most recently the CEO of Myocardia, which is developing personalized and genetically targeted approaches to heart disease. Myocardia was acquired by Bristol Myers Squibb in 2020. Like Pearl, he unfortunately could not be here, but he was kind enough to send along and record his reflections, which we shall see.
Hi, everyone. I wish I could be there with everybody. My name is Tasos Giannakakos, and I am course 10, course 14 in 1994, and it's a real pleasure to be able to talk to you on this really important event, and one that's important to me given how Danny really influenced me as an undergrad at MIT. After I graduated from MIT, just a little bit briefly about me before giving you some thoughts about Danny and what I've been doing. I graduated from MIT in 94, course 10 and 14. I then went to graduate school at Northwestern in biochemical engineering, biotechnology, and went to work in bioprocess R&D at Merck before going to business school at HBS up the road, and then kind of moving to California to help start up a few companies, and I've been doing that ever since. My most recent one being a company called Myocardia, which I was CEO of, and was recently, about a year ago, was sold to Bristol Myers Squibb. But really have enjoyed helping create new companies, and there's been 4 of them, and looking forward to the next one. But I did want to say a little bit about the impact that Dr. Wang had on me at MIT, and really part of the story is just about MIT as a very special place, but really having people like Danny help guide me through those undergrad years left an incredible impression on me. I remember first seeing him as a guest lecturer, and I was assigned to a project with him with 2 of my other classmates, my course 10 classmates, and we were optimizing a bioreaction. And we had to frame our experiments and make sure over the course of a couple of months, we'd write up our conclusions and hopefully get to a point where we're optimizing yield and purity. And I remember walking to his office back then, and many of you might remember these were old army barracks back in the day. Don't look at these beautiful buildings that we have now, but it added a little bit to the air and the aura of walking in to see Danny as, undergrads here doing a big project with him. It was pretty wild. Our labs were down there as well. And I remember one visit in particular where there were the 3 of us. Dr. Wang kindly referred to us as the 3 Musketeers. Once in a while, there's the 3 Stooges, and if you were born after 1995, Google that. We were going in for our progress report at the time. We had sent in a preliminary draft of our research, and what we got back was just a sea of red over some 40 or 50 pages. I had never seen so much red before in any of my work, and it was really shocking. So, I took a long walk home with my lab mates there over the bridge, lived in the Back Bay at the time, and just sort of absorbing it all and kind of taking it in. And I think as the time wore on, what was a super tough experience turned into something really different. I realized he took a lot of care and attention in reviewing that. He gave us an incredible amount of constructive feedback. And I will say, we took the feedback, and in the next month or so, we really leaned into it and kind of took his advice and really improved not just the results. We did a few more experiments, but also the paper. And he couldn't have been more complimentary and really supportive in his response to us after taking in that feedback. So I took a lot away from that experience, and we left that much more bonded to Dr. Wang, but also to each other. And, it started a relationship which, from time to time, he would give us a lot of crap about how bad that first draft was. And we referred to us as the Three Musketeers for decades after that. But, his attention to detail, it was very precise, making sure we ran the right studies, giving thoughtful feedback along the way, and positively reinforcing the effort was really important and stuck with me. I took a lot away from that, including these great memories and friendships. And it was really interesting to see for undergrads, as someone in Danny's position, he took so much care in helping nurture us. And that situation led to more conversations that I had with him, including one that had a lot of practical impact on me career-wise. At the time, this was back in 93, 94, a lot of us chemical engineers had kind of, one of two paths that we would go down. We would either go and work for Shell, some of you may remember this, or the practice school, go to work for Merck. And he grabbed me aside, he didn't even ask me what I wanted to do. He's like, you're gonna go work for Merck. You are not going to go into the petrochemical industry. I just did it, and thank God I did, because it's really kind of put me on a track that's been incredibly rewarding and fulfilling, and that was a big deal for me. So a lot of my experiences with him have really carried into how I have shown up as a leader in various roles that I've had in industry. He was such a huge influence, Danny was. He obviously directed me professionally, but in terms of my style in managing and leading people, paying attention to detail, really connecting with folks, sincerely caring, being authentic about that care with your teams, with your students, and providing feedback. Although at times may be critical, when it's coming from a place of caring, it's really internalized in a positive way. And then when you see those improvements, that positive reinforcement just really puts a lot of wind in your sails, and that's made a big impact to me. He was not a shy guy, and so, being clear and pointed, I appreciate, and I've taken that forward, and I think a lot of folks on my teams over the years, I think they appreciate that directness and clarity from me as well. And I will say, it's obvious by just this gathering how impactful Dr. Wang has been on just so many over the years. And just in these 3 days, he's influenced just so much. In fact, in that group of 3, 2 of us have gone on to be CEOs of biotech companies, and the third is managing like a few $1 billion at an investment fund. And that's just our little group, let alone this bigger cohort that you're hearing from right now. So very rarely do we find people who have made such an important impact. There are a few of them at MIT. There's a few of them in Constantinova, and Danny was certainly one of them. So taking these things into my latest role, maybe telling you a little bit about that. For some of you who are thinking about career choices and how to move from your learnings in Course 10, et cetera, to an industry role, maybe I'll share a little bit. One of the things that I learned at MIT and I've learned from people like Professor Wang have really been seminal in my development. I've been in other institutions, and I have to say that there is just a special place for me in my heart for MIT, just framing problems, learning how to problem solve, getting kind of kidney punched on some exams that I thought I did really, really well on, but really forcing me to learn how to learn and how to approach learning is something that just has really been so valuable to me along the way. In my last company now, where I've been CEO, our focus has been on driving and creating targeted precision therapies for cardiovascular disease. For those of you who have followed the field, cardiovascular diseases are the number one killer of men and women around the world in developing and developed nations. And it's one of the areas that has really lagged behind in terms of new therapeutic development. There's a lot of reasons for that. One relates to our inability to really target the underlying biological and molecular drivers of disease, which forces one into situations where it's expensive to develop these therapies, because without a target, you often get a lot of noise in your clinical trials that drive you to much larger trials. And the corollary to that is the proof of safety really takes a lot of evidence, because again, we're not necessarily understanding the underlying biology that causes things like heart failure, etc. And so we have drugs that we develop that may have unwanted side effects. What we're seeing in oncology and in rare disease, of course, has been really transformative for patients and for companies, where these underlying molecular drivers allow for us to design targets specific to that, and ideally are upstream in the causal disease pathway. So the whole kind of thesis behind MyoKardia, which I joined as CEO about 8 or 9 years ago now, was to bring a lot of that same approach into an area that has essentially been a laggard in innovation, and one where patients are in desperately need of new therapies. We're still, believe it or not, treating these diseases with drugs that have been developed 40 or 50 years ago for things that are completely different and are now being used to essentially ameliorate symptoms, but not changing the course of disease, and certainly not affecting quality of life and outcomes by and large. So that was pretty exciting, to be on a different frontier, to know that if you're successful, you're going to make a big difference. My principles in thinking about this are, first and foremost, science is compelling, and really focusing on doing very high quality science, which is something that MIT trains us all to do very well. At the end of the day, you've got to really be thoughtful about what you're doing in terms of research, the drug discovery. You got to be humble in the process and be aware of the assumptions that you're making along the way, right? And reflect on the fact that those assumptions may be wrong. So always challenge this, always, build organizations and teams that are going to thoughtfully kind of debate these things, because biology is going to throw you curveballs along the way, and you can't do great science without humility and without varying perspectives that you bring together. What was exciting about working at MyoKardia, among many things, was really building an organization of incredible intellectual diversity, background diversity. You've got cardiologists talking to geneticists, talking to molecular biologists, with chemical engineers thinking about these things, and allowing for people to disagree and still respect each other is really how we made breakthroughs. And it was incredible at the end to really see how much energy the organization had, and you really got to places that I don't think even we felt we can get to, but working together we got there. So really fulfilling for me. We're hopefully, fingers crossed, in a few months the FDA approves our therapy, now be in the capable hands of BMS, because there are millions of people out there that really could use treatments like the ones that we're developing. As I think about sort of the direction here, I gravitate towards areas where we can significantly impact society. Often I think about areas that affect a lot of folks. So we're spending time and energy and making great progress in rare disease and oncology, and I think there are many other areas that are hopefully building momentum. Neurological disorders, Alzheimer's, inflammation and respiratory disorders, certainly cardiovascular and metabolic diseases. We just got to do better and more efficiently getting to drugs that are curative or as close to cures as we can get. And that's going to happen from folks coming out of MIT and other places that really are able to dig deep and understand the drivers of disease and the design therapies that address that specifically. Some trends that I expect to continue. I think this theme of precision medicine is really the way to go, and tailoring treatments to specific profiles of patients. What that requires is understanding not only of the underlying molecular biology or molecular drivers, but really being thoughtful about how we phenotype and subgroup patients. Not everyone is the same, and we need to really take a good look at markers. Some of those may be genetic markers, some may be circulating biomarkers, others may be functional and clinical markers. But taking that comprehensive picture and really saying one patient is not like the other, let's understand the differences and let's tailor the drug, the dose, etc., the treatment paradigm to that particular patient. We still have a one-size-fits-all approach in cardiovascular medicine by and large, and that has to change if we're going to really dramatically improve outcomes for these patients. I think part of that, the precision piece, both in terms of the drug side, the underlying biology side, and the clinical profile side, we will see a continued convergence in various disciplines. So what I mean by that is, now we think about, genetics in talking about cardiovascular medicine. 15 years ago that wasn't quite the case. 30 years ago in oncology it wasn't the case, or 25, it is now. We're going to layer that on a little bit more, but I also think we're going to see, and we have already started to see, the power of data sciences and computational approaches, which have to be integrated with the biology. I think it's something by and large that industry is struggling with, whether it's a technology company that is very steeped in engineering and in data science, trying to bridge the gap to the newbies who are the biologists or the biochemical engineers, or the other way around, where it's our life science companies who are pulling in the data scientists, figuring out a way to get those teams to really work together in an integrated fashion. It's going to happen. It's happening at some level in some organizations, but capitalizing on that is going to create a tremendous amount of value, I think, across the entire innovative spectrum, from the provision of healthcare services to the discovery of novel biological targets to cure disease. I'll give you one example of what I'm talking about here, and I'll keep picking on areas that my last company, MyoKardia, was focused on. We have today on our wrist the ability to measure electrocardiograms, the ability to measure step count and activity. In some ways, we can see in places like UCSF and Stanford, and efforts at MIT are using machine learning to translate those readings into measurements that we otherwise couldn't get non-invasively, or would need more expensive approaches like echocardiography to get. Today, we can use ECG and algorithms that are based on ECG to measure things like the dimension of the heart, the left ventricular mass. This is pretty remarkable. We approached the FDA in this particular disease area to get approvals, if we can't on improvements in mortality, on improvements in how patients feel and function. Those are absolutely things now that we can measure on ambulatory wearable devices. So we're now collecting longitudinal information over years, maybe even a decade, on people's cardiovascular health. We can look at improving diagnosis, which is in some instances woefully low, and we catch the disease when it's too far gone and it's too late. Sometimes in sudden death situations is the first time we hear of the ailment in a particular person. But not only improving the screening and the diagnosis, but monitoring the progression of the disease, which allows us to then better understand how to best intervene and how to optimize treatment paradigm and plans for these patients. To me, this is an area that is so critical. If you couple it with biology, we ought to be able to improve outcomes for a large number of patients in a much more efficient way, and discover new biological targets that by definition will be precision in nature and targeted. So this is what my hope is. I'm excited to continue to work in this area where we're bringing together multiple different sciences, and of course at the end of the day, it's all about the people like you who get involved in rallying and developing great teams. Working on new treatments, new diagnostics, and new interventions is absolutely a team sport. I don't believe we have a genius model in this line of work. We can't get there unless we really pull together incredibly passionate people who we support to do just phenomenal science. We're solving really difficult problems. It's a creative endeavor at the end of the day, and every creative endeavor requires that level of intellectual and experiential diversity and humility to pull off just an amazing sort of project and performance. So with that, I want to encourage all of you to take with you what you're learning here at MIT. Let's think also about the legacy that Dr. Wong left us and how he touched folks like me, like my team, the Three Stooges, and like so many of us here, and has influenced not just our thoughtfulness and rigor in conducting our work and our research, but also the caring and attention that he took in developing individuals like me. We'll always remember you, Danny, and I'm looking forward to seeing everybody in the coming months. I hope to make a trip out there, especially talking to the undergrads and grad students. Thanks again for the opportunity to share some thoughts about Danny and the Institute. Bye.
All right. Well, I especially loved hearing about the flooding of the lab. That brings us to our last speaker, Constantinova. I'm sorry, I butchered this man. He received his, Constantine received his PhD in biochemical engineering from Osaka University in Japan. His career in the biopharmaceutical field has included leadership roles at Bayer, Genzyme, Sanofi. Constantine is currently the CTO at Kodiak Biosciences, which is harnessing exosomes, natural intracellular messengers, to pioneer a new class of biologic medicines, exosome therapeutics, with high potential targets in oncology, immune-based diseases, metabolic and fibrotic disorders, neurodegenerative disorders, and rare diseases. And of course, this involves important work in downstream process, which we think would be especially appropriate for honoring Danny's legacy. So thank you.
Thank you so much, and thank you so much for inviting me to this very special 2 days. Has been a really amazing time. I enjoy every single lecture, from the very first one to the very last one. Really amazing. Has been very enriching, both technically and emotionally. In interest of time, given that now it's 5 o'clock, what we heard yesterday, this is about tennis time, and reception 5:30. I'll go ahead with the story here that I have. I would like to introduce to you a new therapeutic modality, relatively complex, biological nanoparticles called exosomes that Kodiak is transforming into a new therapeutic opportunity and new therapeutic modality. In addition to telling you the story of exosomes, what they are and how we manufacture them at large scale, I also want to communicate that the fundamental, timeless biochemical engineering concepts and principles developed by Professor Wang, that you in this audience have evolved even further and will continue to do so, are applicable to complex new modality, ATMPs. And this is what actually we have done in terms of manufacturing, evolve further these principles and concepts of process development and manufacturing. And this is the underlying message I would like to give to the audience. This is let me see now. Am I pressing the right one? Oh, right here. Yes, thank you. Yeah, this is briefly the couple of topics I would like to cover. Just about exosomes and their development by academia and industry today. There is a very interesting dynamic related to what new modality is. And I hope you're interested to hear that. Then tell a little bit about how we manufacture exosomes at large scale under GMP conditions, and take a look forward what is ahead of us in respect to this therapeutic modality. What are exosomes? For those of you who do not know, these are nanoparticles produced by all living cells, human cells, is insect cells, plant cells, bacteria in slightly different shape, and also yeast. They are really in massive amounts in our bodies. In fact, the exosomes at any moment in our bodies, human body, they are more than the cells we have, significantly more, several orders of magnitude. They are known as information carriers between different cell types, different organs, and they act remotely, which is very important. And critical for their application as a therapeutic modality is that they are invisible to the immune system. So they are stealth, really, and this helps tremendously create powerful therapeutic modalities. And they have really broad therapeutic potential, and I will mention some of the opportunities that we're currently exploring. This is how they look like. This is EM picture. As you can notice, they are very diverse in terms of size. Some of them are about 15 nanometers, some of them are larger, up to about 200 nanometers. But when they're purified, they look very nice, round like pearls, and they're pretty uniform. So something interesting that I would like to share from personal perspective that I find very interesting. I have been in the industry for about 30 years. And believe it or not, there is only one time when I work really on brand new therapeutic modality in terms of industrial application, and these are the exosomes. There are a lot of new therapeutic modalities, as we call them, like gene therapy and cell therapies. However, they have been around, and industry has worked on them for about 25, 30 years. And the industry application of exosomes started 6 years ago, basically. So it's really very new. And with this, we actually see tremendous opportunities, but also significant risks. And both academia and industry is working, partnering to systematically address these risks so we can use the potential of exosomes as therapeutics. This field, although very new, is maturing, and here I use one simple indicator. I think it's interesting, basically to track over the years the ratio of industry to academic participation in major conferences. And this is a Congress of the International Society of Extracellular Vesicles, which is the largest scientific body covering this field. And you can see that over the last few years, very steadily, the industrial participation is increasing, which to me is a good indication. This means that the field is maturing. It's interesting dynamics, actually, as what we found when we started Kodiak. In terms of the academic participation and the industrial participation, what we found is that the exosome science, actually this community exists very separately. It's very large, very well organized. They have their own conferences, and it's in fact looks like a separated world. We, biochemical engineers, like most of us here and myself, we have our own conferences, our own events and meetings. And honestly, these two communities, they have been separated. They didn't know about each other. When we started attending the exosome meetings and conferences, we discovered that these people know a lot about exosome biology and biochemistry, but basically don't know anything about manufacturing. And we, in the biochemical engineering community, did not know at this point much about exosomes, but we knew a lot about process development. So obviously, we had to somehow merge and melt these two different communities, and this is actually what we did at Kodiak. Now we unified some melting pot that really works very well in terms of enriching the knowledge and merging the opportunities from both sides. I would like to use the opportunity to take a step back and just share with you something that I read recently, an interesting book here by Professor Stokes called the Princeton Quadrant here from Princeton, who talks about the doctrines in scientific research in the United States. And the story starts many years ago after the Second World War. when actually a very influential person related to MIT, he has been a dean of the School of Engineering, and some of you may have heard about him, Professor Bush, came up with a concept about evolution of science and basic research. And if simply interpreted, with a linear plot, basic research is on one side of the line, and the applied research is on the other side of the line. So two kind of extremes. And in the middle is something that is similar to what we do. 50 years later, Professor Stokes from Princeton try to modify this and make it a little bit more, if you want, collaborative, because from this perspective, it looks that basic research and applied research are pulling in different directions, a little bit against each other. So what was done, he flipped this line here 90 degrees and created this nicely structured quadrant. Here is the fundamental understanding, fundamental research, and this is technology development, and named these quadrants in the following way. The pure basic research is the Bohr quadrant, obviously. The use-inspired research he called Pasteur quadrant, and the pure applied research he called the Edison quadrant. And now, if we think about exosomes and what is happening there, because it's such a new field, all these quadrants are really active at this point, and they need a lot of attention. So academia is working in these two, actually, quadrants, and industrial research in these two. And people like me, biochemical engineers developing processes, are more in what he calls the Edison quadrant. And I'll use this concept again to illustrate some other ideas. So a little bit about Kodiak. The way we evolved the company, we started it in late 2015, so about 6 years ago. We wanted from the very beginning to build a company that has a platform. We already heard from a previous speaker how important it is to have sound platform, but also not to have only a platform company, but also to develop products. So it's both a platform and product company. And I have to acknowledge here the contribution, intellectual and strategic, to Noubar Afeyan, who actually was advising us to take this path. And it was very, very important for how we organize everything. So we have our platform with two arms. One is the drug creation. We develop really powerful technology how to engineer the exosomes. With genetic engineering of the cells or by manipulation after purification. And then the other one is the manufacturing, how we really massively produce these materials, because this has never been done before at large scale. So there is a lot of synergy, obviously, between the two, which is very important. Just to give you a sense of the potential of engineering the exosomes. You can create really very sophisticated constructs that have a tremendous flexibility and therapeutic power. On the right side, these are our clinical material constructs that are in the clinic now, two of them. The third one is on the way. The exosomes can be engineered so that you can decorate them on the surface with proteins or peptides, or inside you can load them with peptides and proteins by engineering the cells, and we're using immortalized human cells. Post-purification, you can load them with small molecules, nucleic acid, and the like, and of course you can combine all these approaches and generate a really advanced construct with incredible therapeutic potential. Just to mention that we're currently working on technology, and we have very interesting data with loading the exosomes with AAVs. And this is really important because in many cases in gene therapy using AAVs, the immune system is reacting, they're neutralizing antibodies, and this is creating problem with the dose. If you can really shield the AAVs in the exosomes which are invisible to the immune system, then probably this problem can be alleviated. In addition to this, the exosomes, we believe, also offer a great potential for creating very powerful vaccine constructs, and this is what we're exploring as well. This is how exosomes, engineered exosomes, look under microscope, and this is actually the wild type of human exosome. And then if the same type of exosomes is engineered, the same cell line, with it's decorated with protein on the surface, you can see this layer here of proteins. This is one of our most important scaffolds that we use to engineer the exosomes on further on the surface with active payloads. In many cases, when we show this picture, people think that this is actually a COVID virus. And it's not COVID virus, it just looks like this because actually structurally they're very similar. So a little bit about the manufacturing. When we started all this, it was clear to us that we have to make a very important decision about the architecture of the process that we would like to design. Maybe some of you are familiar with this graph. It's a general graph not only for biotech, applies to many industries, but it says that in the early phase, when you do architectural design, conceptual design, for this short period of time, you spent only 8% of the total cost of the project. But during this important period, you commit about 80% of the entire cost of the future. So how important is the architectural design in this particular case? And it was not easy because in the beginning, what we inherited was as a way, the standard for making exosomes was ultracentrifugation. This is ultracentrifuge, and it is used very extensively in academia because this field is still very much driven by academic research. And ultracentrifugation does a very good job in academia. You can purify a tiny small amount, very well purified, but small amount of exosomes, which are perhaps enough for academic research. But if you have to imagine how to supply 100,000 and 200,000 patients with centrifuges, then you can think of a large room full of probably hundreds of centrifuges running in parallel. And for biochemical engineers, for engineers, this is a little bit horrifying picture. So at this point, and this was 2016, we had to really decide what to do. Can we really do something special to make the centrifuges really work for us? Or we have to embark on something completely new. And we decided that centrifuge is not the way to go, but what we're going to do is to use really the most advanced biochemical engineering concept principles, evolve them further, the principles that Professor Wang, Professor Cooney, and many of you have developed over the years, and apply this to exosomes and see what happens. So it was a risk because it was not done before, but actually it became very successful, and we didn't look back. So this is how our process architecture at high level looks like today. We use immortalized human cells grown in chemically defined media. We're operating this in very high cell density, 500-liter disposable reactors using perfusion technology, running the process for a month or two. And then we have designed, optimized for exosome manufacturing, sequence, filtration, and chromatography steps, really specially designed for this complex modality. After purification, we can, if the exosomes are already produced by the cells so that they are already therapeutically active, we can go directly to filling. Or if we need to further load them with active therapeutic payloads, we go through another 2-unit operations of loading of the exosomes. So we have developed such kind of loading technology after cleanup, as we call it, and then we do the fill. So that's really how the process of manufacturing of exosomes looks like. This is the indication of our attention to purity of the exosomes, very, very important. We apply mass spectrometry quite a bit to guide process development, and this is actually early case when we had less pure drug substance. This scatter plot here indicates that we are very far from the reference standard. if the scatter diminishes and everything lines up on the 45-degree line here, this is indication for a very high level of purity, and this is what you're seeing in this right panel with beautiful whitish material. And these are highly pure, highly concentrated engineered exosomes. Now, we scale up this, and we produce a couple of different lots for clinical trials, and this is actually some photos of sublots from GMP manufacturing of engineered exosomes. These are sublots from the perfusion run that we purify also. So this is a drug substance, again, highly purified, engineered, concentrated exosomes that are ready to be used either for further loading or filling in vials, depending on the construct. And this is massive amount, so you can imagine that the tiny, small amount of material that is being produced in centrifuges, there is no way actually to make this in industrial amounts. Now, this is good so far, but again, when we progress and we go to the next level and we have to commercialize such kind of modality and supply 100,000 or 500,000 patients with drug, we need to increase the performance of the process even further. So what is ahead of us? Again, solid and sound biochemical engineering principles. We structure the opportunities actually where the hotspots in the process and where we can further increase the productivity and the performance from the beginning, from cell line down to actually purification and the facility engineering. And is our target to actually improve the productivity more than 10x, maybe 20x or 30x. And we are actually on the way of doing this because many of these projects are active and generating very promising results. Just an example. So if you further define the opportunities here, there are a couple of, again, options. The important thing is that we talk about 10x or 20x improvement. We're not really looking for one magical solution, one red button that really creates this. We have distributed this across the process, and somewhere in some areas we have 3x, 2x improvement, which is totally possible. And the good news is that these are not really additive, they're multiplicative. And when you multiply these improvement elements, you get really higher than 10x productivity, and this is really possible. It's exciting, and again, this is because I mentioned this is a brand new therapeutic modality in very well developed and established platform, and old modality, perhaps this would have been very, very difficult. Now, this is again back to the Pasteur quadrant. All these elements are in this area, more applied area, obviously, and this is something that biochemical engineering should tackle, and we'll continue to push in this direction, hopefully with contribution and ideas from the community and the people in this room. However, in order this field to progress further, there is a lot of fundamental academic work that needs to be done. And there are a lot of bullet points here. The list is probably endless. I just defined some of them that really resonate with me. And the first one, for example, is that the exosome synthesis pathways are not understood. These are incredibly complex structures. They're like small cells without actually having a genome. And how their components are synthesized, how they are assembled, remains to be understood. And it's very important because if we do that and if we have control over it, then in this case, we'll be able to even further in more targeted and precise way engineer them and have them do what we want them to do. Again, this using the quadrant that I show several times, this really belongs mostly into the area of academic research and perhaps some industrial research. So I would like to finish with one last slide that is more general, a little bit childish cartoon here, but to illustrate the concept of what we see and what I have seen many times, what is the typical trajectory of really working on complex development projects. And it's not only in biotech, but in this particular case, we're considering biotech. And this is related to the concept of two types of simplicity. So when you start on this journey, in the beginning, very often we don't know how much we don't know. Things look simple, and very often we are maybe even a little bit arrogant in thinking that we can solve the technical and the scientific problem relatively quickly, and we may have a program in the clinic in a year or two. As we get deeper into the research, we're starting discovering a lot of challenges. The life becomes very frustrating. We're entering the cloud of complexity, and this really is a tough time because we come to realize that things are much more complex than we thought. But as you progress more and more and you push, then you slowly start seeing that things become a little bit brighter, and maybe some rays from the sun sometimes are, you can see them already. And eventually, theoretically, when everything really becomes very well understood, you enter another area, what you can call the simplicity beyond complexity. This is when you understand things and the basic principles, and you can really control everything. Now, in biotech, maybe we're never going to be completely here. Maybe we're going to navigate somewhere in this area, but you get the point. And it's important that we don't stop here, and this is really, really critical. So there is a reference here by a poet, Oliver Holmes, who lived 100 years ago. Some of you may know him, who actually says exactly about that. This is a beautiful statement. I would not give much for the simplicity this side of complexity, but I will give my life for the simplicity on the other side of complexity. And I think we all feel a little bit probably this way. And probably this is the right moment to just acknowledge that Professor Wang taught us and left us a legacy that actually gives us the tools and the principles to go through the cloud of complexity and not to give up, and also taught us how to be courageous. So with this thought, I would like to finish, and thank you so much.
Thank you, Constantine. I believe because our reception is actually starting at 5:30, that we're going to encourage people to pursue you at the reception table with their eager questions. And before I thank everyone and in the official symposium, I was wondering if Vicki would like to say any final words. If so, I can bring the mic directly to you.
I'm short. Well, I want to thank you, Paula. Thank you, Charlie. Thank you, Nubar. Thank all the fearsome foursome women who helped put this all together. This, to me, this last 2 days have been a journey down memory lane. It's been poignant. It's been funny. It's been inspiring. I want to thank all of you for coming. It's so great to see so many of you. You know, we've all had wonderful memories. And today was just amazing in terms of what's happening. But what I'm excited about is the legacy that's going to continue on with the fellowship and the innovation for the professors. So thank you again. Thank you.
Thank you, Vicki. Thank you, Vicki. It's just amazing. It just touches me so much to see everyone here and to hear from Vicki like that. And I just want to thank all of you for joining in the celebration of Danny. I wanted to thank everyone who helped to make this possible, especially, as Vicki said, the women behind the scenes. And that includes Gwen Wilcox and Sandra Lopes and Heather Upshaw, who has done a huge amount of work, and Melanie, and Melanie Kaufman, who has done an incredible amount of work. These are the people who were contacting you, organizing things, putting together the schedule, putting together this room, and the reception and the wonderful food, and I really want to thank them. And I also like to thank our organizing committee, Crystal Prather and Charlie Cooney, and thank all of you for participating in this event. It's your presence that has made it so special. It's your ideas and your innovation which are going to bring us forward in this next generation of ideas. And I really look forward to hearing from you as you go through those journeys. Thank you so much for coming to honor Daniel I.C. Wang. Thank you.